Hello and welcome to the live session course on experimental biochemistry. I am Srijan Sen from IIT Kharagpur, and today I will be taking your week 7 live session. So, I guess you have gone through the lectures that were shared with you in the previous classes, as well as you have gone through this week's lecture session and the tutorials as well. Okay, so in any ways I would be discussing in brief with you the lecture slides and giving you important pointers that will be beneficial for your exams, as well as in sorting out what will be the important portions that you need to focus. Okay, right, so I'm sharing my slides with you all, and hope you can get a proper understanding of the class, and then in the end we will be discussing with the question and answers of what we have seen in the lectures as well as what we were taught in this week. Okay, so let us begin with the class. I guess I am visible to you all, right? So I'm sharing my slides with you. Okay, that's great then. So basically, in today's class, we would be discussing with the isolation and characterization of proteins, and this is more of you can understand if you are doing this in the lab, the breaking of the cell, cell harvesting, and all, and dealing with how to take out the proteins from the bacterial cell when you harvest them and all. So we would be focusing on all of this in today's class. So let us first focus on the isolation of proteins and from where can we isolate proteins. So proteins can be isolated from natural sources via recombinant and via recombinant proteins that are expressed from the different sources. So there are the two ways. So from the natural sources, you can get it from the whole organism, tissues, then you have the embryos, tissue culture cells, and microorganisms. On the other hand, there are recombinant protein that are expressed from the different sources. So you have microorganisms from the plants, fungi, then mammalian cell systems from where you can recombinantly express the proteins and then you isolate them. Even you have the Drosophila system that are the insect systems and all. Right. So the general strategy behind this isolation of proteins and protein purification is that this is your the bacterial cell wall, and then this is the bacterial cell. So your proteins get expressed somewhere inside of it, and so the general strategy is that you have a tissue or a microorganism, then you disrupt it, you get the crude fractionation, and then you selectively fractionate them according to the different sizes and all, and from there you can isolate your protein of interest. Okay, so that's all about isolation of proteins from the bacterial system or from tissues and microorganisms. So there are different protein isolation methods that have been stated or are probably known to you all. One is based on solubility, so you can salt them out, salt out the proteins. Then you have the centrifugation, chromatography, where you can separate or isolate proteins based on size, mass, using the gel filtration, which we discussed in our previous class. Then according to the charge, if you want to isolate them, you have the ion exchange chromatography, then you have the hydrophobicity, you have the hydrophobic interactions. Based on that, you can isolate the proteins. You have reverse phase chromatography, then affinity chromatography, and antibody binding and all. So those are the very important or the very essential methods that you should be knowing for isolation of proteins from the different systems, be it from a natural source or you recombinantly do it. But if you're doing it from a natural source, it gets a lot more complicated depending from which part of the nature you want to isolate it. Rather, if you're doing it recombinantly, you have to also handle with the when you're disrupting the cells, you have to handle with the debris, cell debris, and the other cell particles and the cell uh, say organelles and all. Okay, so from there you have to isolate your protein, and then you need to do it, right? Okay. So there is a selective precipitation that you can do based on the ionic strength, pH, organic solvents, molecular crowding agents like polyethylene glycol. So specific property of target proteins, you have the charge, hydrophobicity, and then based on solubility and the size. So we need to understand, like I was speaking of salting out, so that you need to understand what salt fractionation is. So a mild increase in salt concentration, like low ionic strength, the solubility increases, that's salting in. Now when the salt concentration increases or are very high, then what happens, there is a decrease in solubility with the increasing ionic strength. So that's about the salting out effect. So there are two terms which you definitely need to keep in mind. One is the salting in and the other one is salting out. Okay, so that is how it works. So if you have very low concentration of salts in over there, that will also lead to the decrease in solubility, as well as very high concentration also leads to salting out. But with increase in the salt concentration, you will see that solubility increases up to a certain limit, a boundary condition, and then it again starts salting out. Okay. So there are some ions that are more effective than the others in affecting the protein solubility. That can be explained by the Hofmeister series. Like you see, more salting out, like the more the salting out effect, it also stabilizes the proteins, because the protein ultimately unfolds, and that will help you in stabilizing the proteins. Rather, in a similar way of salting in is basically denaturing your protein, so that is also salting in. So less amounts of salt uh, ideally present in the system can lead to the denaturation of the proteins and leads to the salting in effect. So you need to use the ideal conditions of the salts that you want to use in order to stabilize your protein, as well as you should be able to keep your protein in the natured state or its native state, the folded state. You cannot denature the protein. Like for example, if you're using guanidium, the salt that's present over here, it ultimately destabilizes your protein because it denatures your protein. Although there is not much of salting out, but it denatures your protein to a lot more extent. On the other hand, if you see, if you're using a sulfate like a magnesium sulfate something, or a magnesium phosphate or something, that's the ideal condition to keep your protein highly stable in that particular solution or a condition. Right, so the mode of action of the salt fractionation is high concentration of small, highly charged ions such as ammonium sulfate compete with the proteins. Uh, there is not, I'm not sure at which time I would be starting. So probably maybe in 30 minutes or so, just after I conclude with my lecture. Right. So high concentration of small highly charged ions, like you have the ammonium sulfate that competes with the proteins to interact with the water molecules. Water removed from the protein solubility results in the decrease in solubility and ultimately leads to precipitation. Okay, so then what you need to do after probably your protein precipitates out, then you have to remove these precipitates, such that because the more the precipitation due to the Le Chatelier's principle, the entire of the protein starts precipitating out and goes out of the solution. So what you need to do is just remove the precipitants, maybe keep it and then again denature it and lead it to renaturation. You can do that, or maybe just go ahead with the soluble fraction of your protein or so. So that is why it is very important to use the right amount of ionic strength and optimized ionic strength um, with the right amount of salts and the right salt as well, like the ammonium sulfate or maybe the phosphates and all. You have to use it in ideal situations or conditions to keep your protein soluble. Right. Okay. So now let's talk about a very nice technique for that is very important when you are isolating proteins from a very complex system, or from a very um, un, un you can unwanted materials present in your system. So you have to separate out your proteins. So a very important technique, it's known as the centrifugation. So centrifugation can be of many types. You have the cold or the temperature centrifuges, the temperature that maintains the temperature and all. Then you have a fixed angle rotor centrifuge. You have the swinging bucket centrifuge and all. So they're like you see over here. So the swinging centrifuge, right? Swinging arm rotor centrifuge. So in the fixed angle rotor, you have the rotors that are fixed at a particular angle. You put your falcons or your materials in there, and then you go for the centrifugation. Rather, on the other hand, the swinging arm rotor is more flexible. So these arms are flexible. You put it in there, and after during centrifugation, it just becomes in a perpendicular situation with the arm, and then it rotates. So definitely the pellets that gets collected round over here is collected at the bottom. That's giving you a better centrifugation. Okay, so if you see that there are different speeds in which if you centrifuge a cell or something, you can get the different fragments isolated. So depending on the different types of fragments that you want, you can centrifuge it at different speeds. Like during low centrifugation, so you have the cell homogenate over here, and when you go for low speed centrifugation, the pellet contains your whole cells, the nuclei, the cytoskeletons. Now you increase the speed a bit more, and then your pellet contains mitochondria, peroxisomes, lysozymes. Okay, then again you increase the speed maybe to some more extent, and your pellet contains now the small vesicles, microsomes, because now for example, if you require a nuclei or a cytoskeleton, you would be giving a medium speed centrifugation. But if you want to isolate these ribosomes, viruses, large macromolecules, you need to first separate this out. You cannot directly go for a ribosome pelleting with all these ingredients present. Then everything comes, just literally comes down, and you get just nothing. Right. So it is very important that what is the speed of the centrifugation that you want to use for the better to isolate the cell organelles, the different cell organelles and all. So if you see over here that if you are centrifuging at 500 G for 10 minutes, then you increase it to 10,000 G, then 1 lakh G, and maybe even more, you get the different fragments over here. So these are the different homogeneate, then you have the pellet supernatant, and then another speed. So increase the speed slowly by slowly, and you get the pellets from where you can isolate out the cell organelles, right? Okay. Now, with centrifugation comes a very important other term. right? So there are other ultracentrifugation techniques, the finer separation and less convective mixing using the density gradients that you can use sucrose and the cesium chloride. Okay, as well as there are different sedimentation techniques, the velocity sedimentation, equilibrium sedimentation. Okay, so sedimentation, what do we mean by sedimentation is that forces the centrifugal omega square rm, where omega is the angular velocity, radians per second, distance r is the distance from axis, molecular m is the molecular mass, buoyancy is minus omega square r m naught. M naught is the displaced mass, friction equals to minus fv, where f is the frictional coefficient and v is the radial velocity, and we have the diffusion fragments that plays role over here. So the size of the protein molecules, we need to understand the size of the protein molecules. The diameter of a single hemoglobin molecule is around 5 nanometers. The protein molecules can be quite concentrated. For example, the highest physiological concentration of any protein are the crystallines in the island cells that can be more than um, a 50% protein by weight. Fibrinogen is a large rod-shaped molecule that forms a fibrin blood clot when activated. Now, recombinant DNA, to we need the recombinant DNA to purify the protein, so the DNA can be cloned. Then we have the expression vector DNA, right? So this is the DNA, how we put the DNA into this particular plasmid. So this has to be an expression strain of a particular bacteria, say E. coli or something like the BL21D3 cells. There are so many, right? Then you have the recombinant DNA plasmid. They we ligate it using the DNA ligase. You have the bacterial DNA present over here and the recombinant DNA plasmid. So we transform the recombinant plasmid into the bacteria, right? So then we comes to the expression, bacterial protein expression, where you have, this is your bacterial chromosome, okay, and you have the plasmid present over here. So under appropriate conditions and after the induction with an IPTG or maybe an arabinose, depending on what inducible, it's under the control of which inducible promoter you have, then you express the proteins over here, and finally you harvest the cells. So there are so many expression vectors depending on what type of expression you want to do. Like the prokaryotic expression vector, you have the PDEST, PET, and there are so many. Right, then we have the eukaryotic expression vectors like the PPIC, you have the baculovirus expression, PED vector, PED expression and all. And the host systems for prokaryotic, the very common one is your E. coli, you have the DH5 alpha, you have BL21 cells, there are so many like the D H beta 10, you have the super DH5 alphas and so on. Like then you have the eukaryotic cells, the yeast, you have the Cerevisiae, then you have the insect cells for viruses, you have the HEC293, the human embryonic kidney cells for mammalian cell cultures and all. So all of these can be used for the protein expressions, right? So what you do, you transform the bacterial culture, then the next day you get the colonies. Okay, you plate it on some agar medium with a particular antibiotics, kanamycin or ampicillin or maybe dual antibiotics. The way you want it to work, then you inoculate into fresh medium. You give a primary culture overnight for grow it for 17 to 18 hours. Then you give a culture. Then you transfer the culture, a part of the culture, maybe 1%, 10% to um, another a bigger culture like in 100 ml or 250 ml or something, and then you like that's the inoculation into the fresh medium, and then you let the OD reach to zero point 0.4 to zero point 0.8, and then you induce it with IPTG for the protein expression. So depending on what sort of promoter it's under the control of, so it can be IPTG, it can be arabinose inducement like that, and then you induce it. and followed by that, after the induction you grow it at 37 degrees. That's the ideal temperature, or maybe 42 for different time points, maybe 5 hours, some grows for 3 hours, 5 hours, or maybe 10 hours, sometimes 20, 22 hours. You can grow it at different time periods, right, depending on the concentration of IPTG you are using, or the nature of the protein. So, and then after growing it for a certain period of time, you harvest the cells and you store it, followed by the purification strategy. That's a different part of it. So first comes about the growing and the cell harvesting. So what role does this IPTG play? So addition of IPTG induces the expression of proteins, because it's a molecular mimic of allolactose. So you have studied over there. So allolactose is a lactose metabolite. It does not take part in the metabolic pathway, as well as it triggers the transcription of the lac-operon by binding to the repressor molecule, and then the T7 promoter, or the promoter, the T7 polymerase can sit on the promoter and induce the expression of the proteins, right? Then you need to go for the purification of the proteins. That's very, very important, because when you're harvesting the cells, what happens is that there would be so many cell organelles that will also be present over there, along with some of the cell debris, unwanted materials, salts and all. So you need to differentially centrifuge it, go for a salt precipitation, then solvent precipitation, electrophoresis, column chromatography, and finally you want to get the pure protein. So these are the other techniques that you want to use over there. So what does the differential centrifugation do? So it's separation of proteins on the basis of the size, mass, and density. So it's a function of the size of the protein and the speed of centrifugation. It gives us a rough separation. It is basically fractioning. So what you do, you have the suspension of the broken cells that contains your lysozymes, membrane fragments, other cell organelles. You centrifuge it for a certain period of time at maybe 800 G for 10 minutes, and then your nuclei segments. Then you another centrifuge it with a higher speed for maybe 10 minutes or more, and you get as well the mitochondria, the lysosomes, and the peroxisomes. Finally, you start increasing the size, and in this manner you are eliminating the fragments of the plasma membrane, endoplasmic reticulum, and different cell sediments and all. And then comes the cytosol, from where the ribosomes are excreted, and this portion contains your protein of interest. Then what you can do, you can simply dialyze it, and the process of dialization. It's based on the osmotic pressure. It allows you to get rid of most of the salt ions, because, and however, it is impossible to remove all the salts, because it reaches an equilibrium condition, and when it reaches equilibrium there is no exchange between the inner and the outer membrane, right? So this is at the start of dialysis, and this is at equilibrium. Okay, so you have the salt ions in exchange with the outer environment, and this is the dialysis membrane. Okay, right. So after we are done with all of this, then we can go for the column chromatography. So because in these states we cannot do a chromatography or something, because it would interfere with the column, and it would not only destroy the column, but you won't get a proper resolution of your proteins and all, because it would be interfering with the protein, and that leads to a lot of more chaos inside the column. So it is very important to first go for a centrifugation, eliminate all the unimportant materials, and then you go for a column chromatography. So column chromatography is the most common and the best approach to purifying larger amounts of proteins. It achieves highest level of purity and largest amounts, so it requires low effort, lowest likelihood to damage the protein product, and this is a standard method for any pharmaceutical industry and all. So among the different column chromatographic methods that are known to you all, we have the gel filtration, the ion exchange, the affinity, which I already discussed before. Maybe I will be giving a touch on all of these a bit more, and um, that can already do it. But if you see, it's a gel filtration chromatography. These are very beautiful images, the pictorial images that gives you an idea of a lot of things over here. But I need to go into the details of a few of this to give you a better understanding. Right. So let's start with the gel filtration chromatography. So you have the size exclusion chromatography or the gel filtration chromatography, depending on the solvent medium that you want to use. So the molecules are separated according to the difference in the size as they pass through the hydrophilic polymer. So the polymer beads are composed of cross-linked dextran, which is highly and uniformly porous like the Swiss cheese you can see over here. Large proteins comes out fast because they do not enter all the pores. Like the small proteins comes out last because they get stuck in all the pores. So you have to know where the protein is. So you need to first run a molecular weight marker where you have the known weights of all the proteins over there, and then you add your target protein. If you want to purify an enzyme or substrate, you can understand where your protein elutes, so you will get a better understanding of where your protein is. And then, like the last time I said, you can plot a log, a molecular weight versus the distance or a time or volume, and then you can get a better understanding of the size as well as the molecular weight of the protein. Obviously, this also gives you a better understanding of the complexity of the protein as well. Like if you have a dimer or a tetramer or maybe an octamer, like something like that, so it will also elute at that particular position. Like for an SDS page, what you see, even if it's a tetramer, you cannot identify. It comes out as a monomer, but SEC is a very nice method which gives an idea about the polymers or the oligomeric structures of your particular protein, be it an enzyme or anything, right? So ion exchange chromatography, it's separation based on the basis of so it's separation on the basis of charge absorption. So it's a high resolving power. It has a very high loading capacity. It's widespread applicability. Then it's most frequently chromatographic technique for the protein purification. So there are many types of ion exchange. You have the cation exchange and the anion exchange. Like if your protein is in a buffer where your protein is negatively charged, you can use the beads that are positively charged, like the DAE Sephiroth or something, and then your protein gets bound to this positively charged beads, and you can finally elute them, right? So, like if your beads of positively charged DAE cellulose are there, and you have a mixture of positively and negatively charged, then what happens? The negatively charged proteins elutes, binds over here, whereas your positively charged, due to repulsion, elutes faster and then comes out here. It's a very fantastic thing. Like you have the so this is something very interesting. You can do these kinds of experiments if you know that the PI, if you have prior information of about this proteins and all. And another very interesting chromatography is the affinity chromatography. So it's also adsorption separation, in which the molecule to be purified specifically and reversibly. So to a complementary binding substrate immobilized on an insoluble support. So here the purification is 10,000 times or better from a single step. If you have done an affinity chromatography like the Histag like the nickel NTA chromatography, where the nickel binds to the Histag of your tag to your protein, if you see that you get a very pure protein, it's a single band that you get. Most of the bacterial proteins are non-Histag. Sometimes it's possible that your bacterial protein is Histag. That's definitely possible, why not? But more or less you get a single band when you're running it in a gel after the purification through an affinity chromatography. So you have a matrix over here that's inside the column. Then you have a linker like an INT or something, and you have the amino acid, the Histag present over here that binds to your entire protein or something. Like I'm giving an example, a very common example, like the nickel NTA1. Okay, so instead of nickel, you can use talon beads, which is made up of cobalt or something. So those works even fantastically as well. So these are the three very important. The gel filtration, ion exchange, and affinity are very good examples of column chromatography that you can use for the separation of your proteins after isolating them from the cell debris, or okay, or from the other cell organelles. Okay, so this works very well. Now, now you need to get also there are other techniques like the isoelectric focusing that separates the proteins based on their isoelectric point, the pi value. So if you're using a buffer that's higher than the pi of protein, it will be negatively charged and it will be moving towards the anode. Or if your protein has, if you're using a buffer of say that has a pH that is less than your pi, it will be positively charged and it will move towards the cathode. So it's just focusing depending on the pi or the isoelectric point. But for that you need to have a prior information about the proteins. So when you're doing a basic research on solving the structure, function, and dynamics of a protein, you might not have a prior idea of all of these informations. But if you're having it, the isoelectric focusing works very well. Now the other technique is the polyacrylamide gel electrophoresis. Most of you have done it. You have run an SDS page. You have very good understanding about how to run a page and all. There is a stacking gel, resolving gel. You load your samples. It runs through a tris glycine buffer, and you can separate. So what happens over here, your samples are treated with SDS and the tracking dye, so your proteins are all uniformly negatively charged, and that helps you to just resolve your proteins or separate out your proteins just based on the molecular mass, right? So it separates proteins based on the size using the electrical current. Now, great. So you have heard about isoelectric focusing. You have heard about the polyacrylamide gel electrophoresis. Now, doing this separately might also lead to trouble because there can be two or more proteins that are of that can run in the same direction or can be of the same size, right? So for that, there is another technique known as the preparative electrophoresis that uses the 2D page where you have isoelectric focusing followed by SDS page. So first you separate out the proteins based on their pi, you treat it with a particular buffer, okay? Then your proteins first separate out based on either the size or or their pi. So you see a separation might be there is a clubbing of two or more proteins over here or over here. Then you pick up this particular gel and you put it in the SDS page apparatus and then you run it. So you will see that it will start running at different positions. So this lane will try separating out, this lane will separate out, and from there you can understand that yes, so this is my protein of interest, this is the pi, because obviously you have to run a molecular weight marker to get a better understanding of some known proteins, known pi, and that will definitely help you in isolating your protein of interest. Then you stain it, you get a gel like this, and from there you can tell, oh great, so these are some of the proteins of interest. So you know the size, okay? So when you know the size, it becomes much more easy, and also you can tell like, okay, so this is my protein of interest, so it's about pH of 4 or something, so its pH is around 4 or something, and it's of a size of 43 kilodaltons, and that gives you information in two-dimensional methods, and you get an understanding in the two different um, uh, what's called that, two different axes. Okay. Okay, so that's all about the lectures I have. It's not much because we had talked about chromatography in the previous class. We had talked about the gel and all in the previous classes. So you can definitely check back with the lectures and all if you have any doubts regarding this. So this is a very short lecture. So I would maybe give 2 minutes or maybe a 5 minutes break, and then we can start with the practice problems. Okay. Okay, so let us start with the practice problems. Yeah. So the first one, so I think we do not have many people today, maybe some exam or something, right? Anyway, so I see only one person, so yeah, that's good. So if you want to give an answer, you can just put up in the chat box, right? And anyways, I will be telling you the answers. Okay, so let's start. Which of the following factors influence bacterial growth in growth media? Temperature, nutrients, pH, all of the above. So, which of the factors are important for bacterial growth in the growth media that is required? Is it temperature, nutrients, pH, or all of the above? So, first, you people, you can try giving an answer, and then maybe I will tell you if that's okay. If you know it, you can just write the answer in the chat box. You it's like it's not very tough. Okay, so it's very pretty easy, so you can actually so the answer is all of the above because all of these ingredients are important. You need to have an optimum pH, you need to have all the nutrients that are important, the amino acids that are important for the growth of the bacteria. You also need a certain specific temperature that will be there for the growth, and also you cannot grow it at very high temperature or at very low temperature. The bacteria does not grow, so optimum temperature, the right amount of nutrients, and pH, all of these are very very important. Okay, the next one is lysogeny broth does not contain. So lysogeny broth does not contain what? Tryptone, NaCl, yeast extract, or agar. What does it not contain? Maybe if you know an answer, you can give me an answer. Maybe just give it a shot, a try or something. No? Okay. So lysogeny broth contains tryptone, it contains NaCl, as well as it contains the yeast extract. So these are the very three important things that should be present in a broth that is there for the growth of the bacteria and all. So we need all of these three, but what we do not require is agar. So we do not require agar in the lysogeny broth. We only require agar during when we are making agar plates for the growth of bacteria or something. Okay, like the colonies that we have or something. Okay, so agar is we do not contain, the lysogeny broth does not contain agar. Okay. We have the next one. Which of the following elements is a micronutrient for bacterial growth? So there is the macronutrients, there we have micronutrients. So which of this is like is a micronutrient for bacterial growth? Carbon, nitrogen, calcium, cobalt. So if you know that the macronutrients for the bacterial growth are carbon, nitrogen, You have the calcium, but cobalt is a micronutrient. It's not very necessary. That's why it's a micronutrient. Okay. So now coming to a very important question. In order to lyse cells, sonication is used. This process breaks apart the cell by what? Sound energy, heat energy, or light energy. So what is the type of energy that sonication technique uses for lysing the cells? You want to try giving an answer? No, maybe you don't want to try. That's fine. Um, so sonication is something which uses the sound energy for breaking apart the cells. When you will do it, probably if you're in a protein biology lab, you need to do sonication on a very regular basis during the purification of proteins and all. So that will definitely help you. So sonication is very, very important, and that breaks apart the cells by sound energy. Okay, a protein is positively charged when? So when will your protein be positively charged? I just explained it. When the pH is less than pi, pH greater than pi, pH equals to pi, or it does not depend on the pi. So when does your protein become positively charged? Okay, so when your, for example, your PI is say 6 and you have used a pH, when you use a buffer of pH 9, so your pH is greater than PI, so it will be negatively charged. The vice versa case, if you're using a buffer of pH 5, it will be positively charged. So pH less than PI gives you, makes your protein positively charged. Right. In the next question, in size exclusion chromatography, proteins are separated based on what? So we have the surface charge, size and shape, affinity, all of the above. Definitely not affinity. So option C and D is incorrect. The surface charge is also not right. We separate it based on the size as well as shape. Okay, the next one. So you have a mixture of three proteins of sizes 50 kilodaltons, 30 kilodaltons, and 12 kilodaltons. Are separated by size exclusion chromatography. In which order will they be eluted out? So if you look back to my slides, you can understand that the larger proteins elutes faster, the smaller proteins elutes later in size exclusion chromatography. So you have proteins of three different sizes. You have 50 kilodaltons, you have 30 kilodaltons, and 12 kilodaltons. So 50 kilodalton elutes first, then you have the 30 kilodaltons, and finally the small protein that is of 12 kilodalton starts eluting. Okay, so it's in a decreasing order. So first you have the bigger proteins, then you have the smaller proteins. Why is it so? Because the pores are made in such a manner that the bigger proteins need not enter all the pores, so it gets less time, so it takes less time to reach the elution tube. Rather, the smaller proteins are have to enter all the pores and all, and that is why it has to traverse a longer path and it takes a longer time, right? Now the next question is, imidazole can bind to, so what do we use imidazole? Imidazole is a component that is used in the nickel NTA chromatography during the elution of your his stacked protein. So it binds to the nickel, and we use it for elution of proteins that are linked with his tags, and this tag is linked to the NTA column. So NTA binds with imidazole, sorry, the nickel binds with the imidazole, and it ultimately elutes out your protein. So that's a component that we use for protein elution. right? So you have expressed protein in bacteria in lyse cells. So how will you check if your protein of interest is present in the cell lysate? So analyze the lysate by DLS, dynamic light scattering, analyze the lysate by SDS page, analyze the lysate by measuring absorbance at 280 nanometers, analyze the lysate by circular dichroism. So if you see, what do you do? So you cannot do it DLS because it gives you an idea about the complexity. So definitely it's not a purified sample, so you cannot tell about it. Even your lysate has so many other ingredients, the organelles, and so many other debris that are present, so it would not give an accurate measurement at 280 nanometers. And even if you're analyzing the lysate by CD spectroscopy, it will not give you, because it's complicated or complexity in the lysate, because it's not yet purified. So all of these techniques are very well functional when you know that your sample is purified, your protein is purified, and you're seeing only one band in SDS. But when you're running an SDS page, it can give you an understanding, because protein expression means it's an overexpression more than an expression. Okay, so you will be seeing a very deep band, a very bold band, a thick band in your SDS page, because that's an overexpressed protein, and that will give you an idea that your protein of interest is there. The other techniques don't work that well. Fine. So next question, your protein of interest is 90 amino acids long. What will be the approximate molecular weight? It's a very basic question. Okay, so what do you do? So you know that for the average molecular weight of an amino acid, considering all the 20 amino acids, is for each amino acid we consider as 110 kilodaltons. So you have 90 amino acids, so 90 to 110, it's around 9.9 kilodaltons, and that's the correct answer over here. So C, option C. Okay, great. The next one will be, okay, the proteins absorb at 280 nanometer due to the presence of which amino acid? Another very easy question, but there are two options that can be ideally correct. It's tyrosine, tryptophan. You have serine and histidine, so you know that histidine and serine are not the correct options. Rather, tyrosine and tryptophan will be correct. Had there been phenylalanine, even then these two would have been correct, because phenylalanine ideally absorbs at 265 nanometers. So keep these small informations in mind whenever you're answering these questions, because these will be important. There will be so many negative marking, and that can lead to loss of marks more than gaining of marks. Okay, so these are the very important points that you need to keep in mind. Easy questions, but people do so many mistakes right out there. Okay, now the next question is about finding the concentration of a protein. So you're given the absorbance at 280 nanometers, that is 9. You have the molar extinction coefficient, that is 11,460, molar inverse, centimeter inverse. So you have to find out the concentration. Okay, I guess you can do it. You have to use Lambert-Beard's law. A equals to epsilon CL, where L is 1 centimeter, so you can neglect it. You have A equals to epsilon C, so epsilon is given, you have the absorbance, so 9 by 11,460, as simple as that. You can find out the concentration. So 9 by 11,460 is around 0.79 millimolar. Okay, so you will get 0.00, another 0, or maybe 0.00079, so that's 0.79 millimolars. Yeah, right. So option B is correct over here. These units are very important. Okay, so just so here you are given in molar, and here you are in millimolar. So looking at the units are very important. These are the basic mistakes many people do, so I'm just telling you all. Now, for salting out of proteins, which of the following salts is commonly used? So when I was talking about the salting in, salting out effect, I just said for the salting out of proteins, if you see at the Hofmeister series, you can understand that ammonium and sulfate are the ideal candidates that lead to the salting out of proteins. So ammonium sulfate is the important salt over there. Right. So you can get this from the Hofmeister series I showed over here. Okay, so where was it? Yeah, this one. So this slide. So you can see ammonium salting out, and you have the sulfate that's salting out. So this one is for the salting out effects. Okay. So this series gives you an idea. Maybe you can remember this few of the ions. Okay, that can help you. Okay, so where were we? Okay, the next one. To determine the size of a protein, which of the following techniques is used? Now coming to the size. Okay, so for determining the size of the protein, which of the following techniques can be used? It's a very easy question, but you know what happens over here? Many of them will say this cannot be used for getting an actual size or something. And I don't have to think in that manner. Size exclusion chromatography works fantastically over here. You can get the size when you run the molecular weight marker and something. Even that SDS page is also great over here. Okay, and even analytical ultracentrifuge gives you an idea about the size of the protein. Okay, so all of the above can be used for determining the size of the protein. Okay, right. So, okay, so you have a protein sequence over here. So mention which of the following statements is true for the above protein. The molecular weight of the protein is of size 10.5 kilodalton. The pi is 10. Molar extinction coefficient is something, and it will have a maximum solubility. Okay, so maybe this is not the type of question you can expect in the exam, because for this you need to take the help of prot param tool, okay, which will give you an idea, better idea of the parameters of the protein. So you have the XPC prot param tool, which in which you can just paste the protein sequence and it will give you all of this idea. But what I can do over here is I can give you the informations present over here and ask you the second question. So the protein in the previous question in the buffer of pH 8, okay, and then you can be asked that it can be purified by. Okay, so I'm giving you the informations. So the molecular weight of the protein is 10.5, the pi is 10, and the molar extinction coefficient is 22.4. So all of the three options are correct. So these are the known informations for the particular protein. Now, the protein in the previous question is in buffer P of pH 8. It can be purified by size exclusion chromatography, cation exchange, anion exchange, affinity chromatography using nickel um, 2 plus column. So if you look at it, it's not very tough. Um, it's very easy. If you look at the sequence of the protein, you will get to know that it cannot be purified using affinity chromatography because it does not have a his tag over here like this. Okay, so it does not have it. So you cannot use affinity chromatography. Now you're using so the pi of the protein is 10, and you're using a pH of 8. So your protein would be negatively charged because the pH is sorry, so it will be positively charged. Okay, so it will be positively charged because your pH is less than your pi, so it will be positively charged. So you can do cation exchange chromatography as well as size exclusion chromatography will also work fantastically for this particular sequence. Okay, so both works well, right? I guess you have got it because you cannot use an anion exchange. Okay. Right. Okay, the next one. Which of the following techniques is best suited for estimating the overall type of secondary structure? Okay, so to get an idea about the secondary structure of a protein, um, you have to use CD spectroscopy that gives you the percentage of alpha helix, beta sheets, Even the loops and turns, you will get an idea of using the CD spectroscopy, which you will not get using SDS, gel filtration, or even DLS. DLS gives you about the complexity of a molecule, the diameter of the protein and all. Okay, then the gel filtration is the size, and even you get the size over here. Okay, the next one, the three-dimensional structure of a protein can be determined by. Okay, so now you have the three-dimensional structure. You know that very widely used techniques is the NMR spectroscopy, the X-ray crystallography, that works very well for the second 3D structures. For 2D, you have the CD spectroscopy, and the size exclusion chromatography gives you about the size and the shape. Okay, great. So A and B both are correct over here. Okay, the next one, particle size and sample homogeneity can be determined by. Okay, that's great. So particle size, so when you talk about the particle size and the sample homogeneity, get an idea, you can do it using the DLS method, the complexity and all. You can do using that. You cannot have an idea using SDS page, but you can get it using analytical ultracentrifuge as well as size exclusion chromato. Yeah, you can do using even size exclusion chromatography as well. Yeah, because the shape, even if there is a change in the shape, there will be shift in the peaks and all. So when you do these things in the lab, that is actually very good, because sometimes it's difficult to explain, but otherwise it works well. Now finally, a protein without any affinity tag is being purified from a sonicated crude lysate. Which of the following is the best procedure? Na2 plus column, ACC and exchange salting out, ion exchange nickel column, ACC salting out, ion exchange ACC. So which of them works good? So if you see that options A and C are wrong, because your protein of interest does not have any tag, so you, or a histidine tag, so you cannot do it. Even option B is wrong because the sequence or order of purification is wrong. So the crude lysate obtained after the sonication should be subjected to salting out. By these, proteins which precipitate out are removed, and then you separated the proteins which remains in the supernatant or the solution. Now this is then subjected to an exchange chromatography, and which can separate some contaminants, and then you put it in the size exclusion chromatography and you get an idea about the size. So only then only your protein of interest is present. The other things just is eliminated in the ahead of the steps. So option D is the correct answer over here. Okay, so that's all then I have from my part. If you have any questions, you can definitely go ahead with it. I will be happy to answer them. If not, then it's fine, you can just drop me a mail or something. In any ways, this video is being recorded, so probably the NPTEL team would be sharing with you the slides as well as my recorded lecture. Great then. No questions? Try solving these questions, try solving the assignments, because don't leave it for the last date and all. So try to go through the lectures. If you have any problem, just ask me in the next class. Okay, and I would be very happy if you are studying and giving me answers even in the class. Okay, so that's all then. Thank you then. If you don't have any questions, we will again meet in the next week on Friday at 6 p.m. Yeah, okay. Thank you. Thank you for joining.